Okay, for chapter 10 here, exercise 10, mostly what I want to do here is just kind of clarify some of the things your lab manual talks about. They do a really great job of talking through each of the enzymes that we're testing for here. And there's pretty good pictures in the text as well, a nice table that tells you what you're looking for in order to read the results. So I would refer mostly to the lab manual. I just do want to point out a few things, again, just some clarification. All right, so our exo enzymes, these are all enzymes that are secreted outside of the cell. And certain organisms are going to be able to produce certain of these enzymes, other organisms can't. So we see a variety in organisms, and so again, this is one more way that we characterize organisms and helps us to identify them as well. Of course, all organisms have endoenzymes, or enzymes that work inside of the cell. Those are necessary for life to be sustained. Again, exoenzymes secreted outside of the cell. All of the ones that we're going to be looking at closely here are necessary to hydrolyze some macromolecule outside of the cell. So remember what your macromolecules are: your carbohydrates, your nucleic acids, your lipids, your proteins. So big molecules broken down so that the smaller parts can be used inside of the cell to build cellular machinery or to use as a source of energy. All right, so the first one is a protein. Just remember that's a protein. It's one of the major proteins in milk, casein. And the enzyme we're testing for here is caseinase. I love it when the substrate casein is in the name of the enzyme. That helps you remember what's taking place here. And if you have an enzyme that's chopping up a protein, what kind of products do you expect to get? Well, it shouldn't be too difficult. You're looking for shorter chains of amino acids. Okay, so if you think about what proteins are made of, that's what you're going to get from an enzyme that chops it up. Okay, with all of our inoculations on plates today, we're going to practice our 4-quadrant streaking technique. So it might have been a few labs since we've done this. Remember, first quadrant, pretty heavy. Second quadrant, make sure you're flaming in between there. So we're trying to spread out our organism, trying to get isolated colonies. And it's not really crucial that you get isolated colonies for these tests, but we want to practice that technique. All right, so streak it out, 4-quadrant streak plate on your skim milk agar. This is a agar that looks kind of milky, so shouldn't be too surprising considering what we're testing for here. A positive result is just a clearing in that opacity of the skim milk agar. Okay, so something that's opaque means that you can't see through it. All right, so it's cloudy enough that you can't see through it. Again, there's a really nice picture there in your book that shows what a positive result looks like. Nice clearing and opacity anywhere that there's growth. So anywhere around the colonies. All right, that one's really nice and really very easy to read. Our second enzyme, we're testing for the production of the enzyme that's able to break down gelatin, and that's gelatinase. Gelatin is another protein. It's derived from the hair, hoofs, horns of animals. So it's a structural protein, connective tissue protein. An enzyme, some organisms can make an enzyme that breaks that down, gelatinase. So again, we're looking for shorter chains of amino acids. Okay, so this is the only test that we're inoculating today in a test tube. That gelatin is Your book calls it semi-solid. You can turn the test tube upside down. It certainly looks solid, but it's fairly soft. So when you inoculate, you're just going to stab your loop down in and then come straight back up again. And then what we're looking for a positive result, after we incubate that for a day or so, is the liquefaction of that gelatin. Oops, gelatin. All right, so one caveat. So this usually, for a positive result, it's usually just a little bit runny up at the top here. Usually the whole tube doesn't liquefy. We just don't get that much growth from our organisms that are positive. But there's one caveat here in reading the results, because gelatin is going, boy, this shouldn't be that hard of a word to spell. Gelatin, when the temperature is less than 25 degrees Celsius, is going to be solid. So 25 is just a smidge warmer than room temperature. So when we inoculate these, that they're solid, but above 25 degrees Celsius, then the gelatin is going to be liquid. All right, so when we put these tubes into the incubator, they're all going to liquefy. That's the main reason that gelatin wasn't an acceptable solidification agent to add to our nutrient broths, because even though they were solid at room temperature, when we incubate them to try to get our organisms to grow faster, then it liquefies. All right, so agar has a much higher melting temperature, 100 degrees Celsius, where again, agar starts to liquefy above 25. All right, so because all of the tubes are going to be liquid in the incubator, you have to, in order to read the results here, you have to cool to at least room temp. You might want to make sure, put it on some ice for about 2 minutes, just to make sure that's cooled down enough. And if it's still cool, if it's still solid after you cool it down, then you can call it negative. If it's liquid, when you still cool it down, then that's going to be a positive. Now, in reality, when we get our tubes out, when you come into lab, they will have been in the refrigerator. So we don't need to worry about putting them on the ice, but it is something you have to keep in mind. You can't pull these right out of the incubator and say, oh, they're liquid, it's positive, when in fact all the tubes will be. All right, next up here, we're looking for an enzyme that breaks down starch. It's an enzyme that we have in our saliva as well, alpha amylase. Okay, remember starch is a carbohydrate, and it's actually made of many, many molecules of glucose put together. So if we're chopping up that starch, we're looking for short chains of glucose. That's called dextrin. Maybe some maltose, which hopefully you remember that's a disaccharide made of 2 glucose units, and maybe some individual glucoses as well. All right, so again, we're hydrolyzing that starch. We're going to inoculate starch agar. 4-quadrant technique there. Once again, this is the plate that looks kind of like nutrient agar, so you might want to just label that so you remember what that is. And this is the only test today that we need to add a reagent to after we incubate. So after we incubate, we're going to add iodine. You can just use your grams iodine in your bench. Just flood the whole Petri dish with that iodine. You want to cover up anywhere there's growth. Iodine actually will kind of stick in those helices that a intact starch molecule has, and anywhere that there is starch, it will really turn kind of a really dark brown. So it turns dark brown in presence of starch. But what we're looking for, can the organism make the enzyme to hydrolyze starch? So for a positive result here, we're looking for areas around the colonies where that dark brown color has cleared. Okay, again, I'll refer you, there's a really nice picture in your book that shows this. If it's that dark brown right up to the colonies, then that's a negative. But if you've got a little change in the media around the colonies where that dark brown has disappeared, the iodine is kind of a brownish yellow color. You might see a little bit of that, but you'll see a lessening in that dark brown. Okay, lipids are hydrolyzed by enzymes called lipases. And we'll just make this singular lipid lipase. Lipids are made up of glycerol and fatty acids. So glycerol, this is a 3-carbon molecule, and those 3 fatty acids that would kind of stick out from that. Organisms that make lipase are going to do so to get lots of energy out of it. So the glycerol can be metabolized. Fatty acids are metabolized by beta oxidation. You might have talked about that in lecture. So again, that's an advantage to the organism. And also organisms that produce lipase, it's actually beneficial to them in establishing an infection in the host, especially in places like where our skin can be kind of an oily place. So some organisms that make lipase, that gives them an advantage in terms of invasion. Okay, our media here is the one that's not intuitive. It's called Spirit Blue agar. And it's the one that, if you look at it really closely, it is blue, but it has all these little fat droplets in it. So if you hold it up to the light, you really can't even see through that. So again, your book says to inoculate it kind of in a straight line like this. Either you can inoculate that way or just stick with the 4 quadrants. Either way, this one is a little bit more difficult to read the results. We're going to look at this really carefully. But what we're looking for a positive result is a clearing in the turbidity around the colonies. So in other words, we're looking for those little fat droplets to disappear. So you're able to see through the media in just a little zone around the colonies. Some of your plates won't be blue anymore after you incubate them. That doesn't have anything to do with whether it's positive or not. We have to see a little bit of this clearing. When you hold it up to the light, it helps because the light can come through a little bit easier. Okay, and lastly, another blue media, we're looking for the enzyme that hydrolyzes DNA. We call it DNase. And remember what monomers make up the polymer DNA. We're looking for the liberation of nucleotides. All right. So remember, a nucleotide is the nitrogenous base, the sugar, deoxyribose, as well as the phosphate. So those 3 things make up a nucleotide. All right, the media here, we just call it DNase media. So not because there's DNase in it, but because we are testing for the enzyme DNase. It's another blue one, but this one, if you hold it up to the light, you can see through it. And for a positive result, we're looking for kind of a purple, sometimes more pinkish color change around the colonies. And the reason that we get that blue color in the media comes from a dye called toluidine blue that we put in there. That toluidine blue reacts with the phosphates that are liberated to give this purplish, pinkish color change. And the picture in your book definitely looks purple. It depends on the organism how that turns out. Okay, so again, your book does a really good job of covering that. I just wanted to kind of clarify those reactions. You should know the substrates and the products, as well as the enzymes that we're testing for, the media that we put it on, and how to read the results. Okay, thanks for listening.